I'm just going to dim the lights so everyone can see the slides. Don't fall asleep. Thank you for the introduction, Alexi, and thank you to the assessment committee for being here, Dr. Agegi, Helveg, and Olson, for taking the time to read the thesis and be here today. I like to use the analogy of Pandora's box for my thesis work because once you start asking questions about exposure to chemicals in consumer products, you realize that there's a whole lot more questions that you need to answer. I also like to use this analogy because if you've ever brought home a box straight from the store and opened it up, you might be able to smell some fresh chemicals, and maybe you leave it out on the balcony for a few days. But in any case, this is a very nice analogy to start the discussion today, or my presentation today. So to go over briefly what I will be discussing today is I will be looking at, or I'll be introducing the background and the research needs that I addressed through this thesis work. I'll be going over the methodology, that's how I address these research needs, specifically defining a metric, developing a framework, reviewing models, developing models, and then applying in life cycle assessment and beyond. The presentation will be split into two parts where I'll focus on cosmetic modeling and also food packaging modeling separately. So I'll go over the methods and then the results for the cosmetics and then the methods for the results for the food packaging later on. But first I'll go over these first three bullet points. And really the developing of the models was the focus of my work. And then I'll leave you with some of the main findings and the outlook. In all, this thesis work led to 10 scientific articles and 1 book chapter, and these will be highlighted in blue throughout the presentation. Instead of boring you with all their titles, I did a word cloud, and we can see that this actually captures quite well that my thesis was about modeling consumer exposure to chemicals in consumer products. Actually, consumer products doesn't really appear up there very well, but yeah, you can get the general idea that this is about exposure to chemicals in consumer products. 3 of these articles were first authored, and these were really the models that were predicting exposure to chemicals in cosmetics and food packaging, as I mentioned earlier. So for those of you who are not familiar with life cycle assessment, I'll just go over it briefly. Life cycle assessment, or LCA, as we call it, is an environmental sustainability assessment method that's product-oriented. It's usually comparative, or almost always comparative, where it's either comparing one product with a functionally equivalent product, such as a glass and a plastic water bottle, or it can be comparing different stages of the life cycle of one product. Here's a schematic that I presented in the thesis, that I put together for the thesis, of this general life cycle concept. So up at the top, we have manufacturing. This is the life cycle of 1 can of canned corn. So to start at the top, we have the extraction of the raw materials, the corn and whatever materials are needed, for example, for the can, and then the manufacturing or production of the product, the distribution to retail and to consumers, consumption of the product, and then disposal of the product. In general, life cycle assessment takes into account the human exposure to chemicals that are emitted to the environment from all of these life cycle stages. Life cycle assessment, unlike other assessment approaches like risk assessment, tends to look at best estimates or average scenarios and not worst case scenarios. It's a quantitative and strives to be a consistent method based on 2 major methodological points. Life cycle inventory, which is a list of chemical and mass flows and also energy flows, in general flows that are being extracted from the environment and being emitted to the environment, and life cycle impact assessment. And there's 3 main areas of protection that are covered in life cycle assessment: ecosystems, natural resources, and human health. And human health is the focus of my research. And again, if we consider typical life cycle assessment methodology as applied today, it does not include exposure to chemicals in consumer products. It only includes exposure to chemicals that are being released to the environment through consumer product life cycles. So paradoxically, life cycle assessment is supposed to be looking at the human health impacts of a product. And we all know that using consumer products can actually influence our health and our well-being, or impact our health. And this is kind of the elephant in the room that really my thesis work is addressing. This idea that when we use consumer products, we actually can have this can influence impact our health. And specifically, I'm looking at this with respect to chemicals and consumer products. Just to go over briefly some definitions to make sure we're all on the same page. When I say the word exposure, it's when a chemical is coming in contact with a human. So this can be through inhalation, ingestion, or dermal contact. Exposure is not equivalent to risk. So just because we're exposed to chemicals doesn't mean that there's actually a risk. A risk is a probability, an increased probability of a disease case, or the probability of a disease case. So being exposed to chemicals doesn't necessarily increase our risk of any diseases. So these two things really should be considered separately. Secondly, safety is not equivalent to there being no risk. Risk managers, for example, can set the safety limit for an allowable amount of risk. This might be one cancer case in 1 million people occurring with a probability of 5% or less, or something like this. So it's really important to realize that safety isn't necessarily no risk, but it can also be the absence of measurable risk. Lastly, human toxicity impacts, which is what we call impacts in life cycle assessment due to exposure to chemicals and consumer products, are not equivalent to risk. They're not measuring probability of disease cases. They're an extrapolated indicator of potential toxicity that's being extrapolated from animal toxicity experiments. To put the context of my thesis work kind of in a little history here, we can start back in the late 1990s when the United States government decided that they should be monitoring people in the population for chemical exposure. So they were selecting representative groups, or supposedly representative groups of the population, and measuring chemicals in their urine and their blood. And this started in the late 1990s and is a continuing program today. This is called biomonitoring. Following interest in biomonitoring and the idea that there's thousands of chemicals that can be in consumer products and thousands of chemicals that can be emitted to the environment, life cycle assessment also became interested in including exposure to chemicals that are emitted to the environment in the assessment framework. The basis of this really started with the intake fraction. The intake fraction is a ratio. It's the measure of the mass of chemical that's taken in by all exposed persons versus the mass of a chemical that's emitted to the environment. This is a fraction, and so it's a ratio. So if it's equal to 1, that means all of the chemical that's emitted to the environment is being taken in by people. But this is very rarely the case in terms of environmental emissions. Following this, interest in exposure, there's also been interest in toxicity. And how do we understand the toxicity of thousands or even up to 100,000 chemicals that are in commerce? And again, as Peter pointed out in the presentation yesterday, this number is arguable, but let's say about 40,000 chemicals in commerce. How do we understand their potential for toxicity? This was being addressed in the ToxCast program, also through the United States government, through the regulators. And one of the most novel aspects of this program is the generation of bioassays, of high-throughput screening bioassays. High-throughput screening just refers to the idea that they can process hundreds of different chemicals, or dozens to hundreds of chemicals, with relatively quickly and with high efficiency. And what these assays are doing is, instead of doing experiments on rats or mice, for example, they're using test tubes and enzymes and proteins and other types of biological, basically aspects of a biological pathway that can be altered by a chemical. So when a chemical comes in contact with this, it can elicit some kind of response, and this can be measured relatively easily. Let's see, the other things I would like to point out is the use-tox model, which is a consensus model used in life cycle assessment. It is a model that includes human exposure estimates, again, based on this intake fraction, and it also calculates potential human toxicity impacts or human toxicity impact scores. This model is really the basis of human exposure in life cycle assessment. But there's been some recent work, well, really starting back in 2009 with Stephanie Helvig's work and the UNIP CTEC Life Cycle Initiative, that's the United Nations Environment Program Society for Environmental Toxicology and Chemistry Life Cycle Initiative Program, and looking at indoor exposure and really in occupational settings mostly. Also work on pesticide residues by Peter Vanke. And then in 2013, an interesting thing happened. The use tox model was applied in a high-throughput risk-based, well, it's a high-throughput calculation of exposure to chemicals that are emitted to the environment. And this was an intersection of a model that was made for LCA in a different science policy field. So this was really an interesting turning point, and really I think it was that turning point that led to my thesis project. So this began in 2013, and it was the idea that we need to start developing methods suitable for life cycle assessment, but also for other science policy fields, risk screening and prioritization, for example, where we can look at exposure to chemicals and consumer products as well as exposure to chemicals that are emitted to the environment. And the rest is history and will be what I discuss in the rest of this presentation today. So to start out with the methodology, my work contributed to the development of a new metric. And this metric, like the intake fraction, is called the product intake fraction. And again, it's a ratio. It's a ratio of mass. It's the mass of chemical taken in by exposed persons versus the mass of chemical in a consumer product. And again, if this ratio is 1, or if this fraction is 1, it means all of the chemical in a consumer product is taken in by exposed persons. We were lucky to be featured on the cover of the Environmental Science and Technology journal with this article, and this really started, it gave us a language and a metric to start talking about consumer exposure to chemicals and consumer products in life cycle assessment. And following this metric, we developed a matrix framework to calculate the nerd joke, but to calculate the metric. So this matrix framework is comprised of different compartments. And in general, the modeling, for example, in use tox is compartmental modeling. And what this means is if we can think of the environment, for instance, this room, as a bunch of different discrete zones. So the air in the room, for example, could be one compartment. All of our skin could be another compartment. The tables in the room could be another compartment. And all of these compartments can contain, release, or degrade chemicals. So chemicals can transfer in between these different compartments based on their physical chemical properties and based on aspects of the compartment. So the difficult part is, okay, now we have this matrix framework, but we don't actually know how to estimate all of these different transfers. And that's where my work came in, to develop models to estimate these transfers. So we reviewed all sorts of different models that can be used in this framework, a pretty extensive review led by my colleague Lei Huang. And she looked at all sorts of different transfers that can happen, for example, from solid products or flooring materials. And my contribution to this work was looking at models for cosmetics and models for food packaging. And very specifically, I found that there was no model suitable for estimating exposure to chemicals in cosmetics that considered multiple pathways. And by a pathway, I mean different ways that the chemical can go from the cosmetic to the human. So for example, the chemical in the cosmetic can go into the skin, or it can volatilize into the air and be inhaled. And I couldn't find any model that was suitable for these high-throughput assessments, high-throughput computations that looked at this. I also didn't find any models suitable for food packaging material. So these were really the two focus products. To go into a little bit more detail on the cosmetics, I promise this is the only slide with equations on it, first of all, but let's go through them a little bit. Previous work was based on the topmost equation. The model that I developed is based on the bottom equation. And really the two Okay, let's start with the top one. On the left side of the equation is the mass taken in to a human, in this case into the skin, versus the mass of a chemical in the cosmetic product. It's a function of the rate of transfer from the skin surface, into the skin, and time. In order to operationalize this model for life cycle assessment and to consider multiple pathways, first of all, we determined the best QSAR model to estimate this rate of transfer from the skin surface into the skin. And second of all, we updated the model with the transfer to air. And that's why the bottom model contains a new term, which is the KPA term. And this term is able to take into account the transfer of a chemical into the air as a competing pathway. So you can imagine if you have a chemical on your skin in a cosmetic, it can be volatilizing and going into your skin at the same time. So these two additions, determining the best QSAR and updating this model to include transfer to air, they're novel, but really the most important part of this is that the transfer to air allows us to complete the full mass balance of fate and transfer of a chemical in a cosmetic. And really, my models were focusing on this yellow region with all the different transfers that can occur while you're using a product. And then models like UseTox, for example, can estimate exposure to the population when this cosmetic is washed off, for example. So you can imagine that we're exposed to chemicals in cosmetics that we're not actually using. Maybe some other people are using them. They're being washed off, being transferred to water. They can get into drinking water, and then people can be exposed. And when I say mass balance here, quite simply, it means that all of the different fates of the chemicals, when they're all summed together, they equal 1. So we can now have a full picture and account for all of the different transfers and fate of chemicals in a product, specifically in a cosmetic product. The model very specifically measures intake into the stratum corneum, which is the outermost layer of the epidermis, and this is the layer of the skin that's considered for exposure assessments. It's supposed to be the rate-limiting step for exposure, which means once basically the rate at which it's going into your body is controlled by this layer, and that's the outmost layer of the skin. It's a function of physical chemical properties, very specifically molecular weight, which is a proxy for molecular volume or molecular size, which determines how fast a chemical can move. And also a function of the octanol water partition coefficient, which is a proxy basically for solubility or lipophilicity. It's the ratio of lipophilicity to solubility, basically, the amount of chemical that can be transferring to octanol versus water. And the surface area of the cosmetic that's applied and the amount applied, which contributes to the thickness of the cosmetic on the skin. And then, of course, the time on the skin. When we apply this model, it gives us the opportunity to look at a bunch of different chemicals with different chemical properties and understand or basically how these properties can influence exposure and also their fate. So here I just am looking at 3 different chemicals: isopropyl alcohol, benzyl benzoate, and propylene glycol, which are in a variety of cosmetics. In this particular example, it was just shampoo, and that's the gray line. Here is the 4-minute exposure time for shampoo. This on the x-axis is the exposure duration. This is 10 hours here. And on the y-axis, we have this new metric, the product intake fraction, or the PIF. The top graph is dermal exposure, so the product intake fraction due to intake into the skin. And the bottom graph is the product intake fraction due to inhalation. And there's some pretty cool things that we figured out by applying this model. First of all, if we look at isopropyl alcohol, which is coming up in kind of an orange color here, the magnitude of inhalation of isopropyl alcohol is higher than the magnitude of dermal exposure. And this was quite surprising because in past assessments, it's been assumed that the worst case scenario for exposure to cosmetics is through the skin. But actually here we're seeing with this particular chemical, because it's quite volatile, that the exposure through inhalation was actually higher than the exposure through the skin. Also, we can take a look at, for example, propylene glycol. This is the linear line here, the green dotted line. And why this one's behaving differently than the other two, or why it looks linear, is simply because we haven't gotten to the time where it would saturate out here. So it's moving quite slowly. It's moving into the skin slowly. It's moving into the air slowly. Also, the great thing about this finding that inhalation could be higher than dermal exposure was that recently there were some experiments that also corroborated this in the laboratory. So this has really been a recent topic and concern related to exposure to chemicals and cosmetics. We also found, perhaps intuitively, that exposure to chemicals in cosmetics is higher for, generally higher for products that are left on the skin for a longer period of time. These are generally called the leave-on cosmetics versus the cosmetics that are washed off quickly, such as soap or shampoo or these types of things. It's also important to note that this was the first application of the product intake fraction, and we realized quite quickly that a high product intake fraction does not necessarily mean a high intake. And this is because, let's say, for example, that the product intake fraction is equal to 1, or that means 100% of the chemical is being taken in by the user or by people in the vicinity. This does not necessarily mean that in terms of mass, it's a high amount of mass being taken in, because the concentration in the chemical can be very, very low. The concentration of the chemical in the cosmetic can be very, very low. So sometimes you can have trace amounts of chemicals, for example, 0.001% or very low fractions. Concentration as in a weight fraction, I should specify. Okay, and just to extend that this model is also looking at different pathways. On the top here, we have the washed off fraction, and then on the bottom graph, we have the ventilated fraction. Again, on the x-axis is the exposure duration. And on the y-axis is the post-use fraction emitted. So this was the first time that there was a model looking at the amount of cosmetic that can be emitted after it's being used. And you can imagine that this is important for all sorts of different types of scientific work. For example, looking at the amount of emissions that could cause ecotoxicity. We also found, also quite surprisingly, that environmental exposure, or exposure through environmental pathways, can be of similar magnitude to exposure in the near field, or that means indoors. But this only tends to occur, at least in the examples that we looked at, when the exposure in the near field is very, very, very low. So this means that almost 100% of the chemical is being emitted to the environment, and then people can be exposed this way. So I was very lucky to be able to apply this model in several different studies led by various scientists, some at the United States EPA and some at the University of California, and also in kind of a case study in a book chapter that I co-published with Peter Fanke, or is actually submitted. It's not quite published yet. And this figure here shows just kind of a play on using this model and what it could mean for life cycle assessment. So if you talk to a practitioner, someone who does life cycle assessment, and ask them how they consider environmental emissions after using a cosmetic, they might tell you that they assume 100% of the chemical is being emitted to the environment, or that 10% of the chemical is being emitted to the environment, no matter which chemical and no matter which cosmetic in many cases. But when we apply the model that we developed, we actually see that this is not true. For example, if we look at D-limonene as an example, the left-hand side of this figure is this flat fraction, so just assuming 10% is being washed off. And on the right-hand side of the figure is what the model would predict. The model would predict that 20% of this chemical is taken in by the user, that 80% is volatilized during use, and that actually less than 1% is being washed off to the environment. This results in ecotoxicity impacts that are orders of magnitude less than would be otherwise calculated. Now, I don't want to send the message that people should use my model because it makes ecotoxicity impacts look less. And in general, that is a very not the right message at all. But this example just happens to show that. But you could imagine in a different example, it could be perhaps that 100% is washed off. For example, that propylene glycol scenario. So in that case, the ecotoxicity impacts would be about an order of magnitude higher than assuming it was only 10% that was washed off. The next exciting thing that we learned with using the PIF metric was that when we combined PIF with other data, for example, on co-occurrence of use, so using all sorts of different cosmetics, we could actually predict the exposure that would lead to these urinary concentrations that are being measured in the United States surveys. So we can actually predict actual exposure that's happening in an entire population. In that specific scenario, it was looking at parabens, and parabens are one of the few chemicals that have basically only one use. They can occur naturally in some food items like blueberries, but they are mostly used in cosmetics. And so we were able to actually look at how our model could predict real exposure. With other chemicals, they're used often in a variety of different types of products, so it's really hard to use one model to try to predict any kind of exposure. It's basically impossible in a lot of ways, unless you have the list of all the different products that are being used, how much people are being using them, the concentration in the product, and so on, and the models developed for all these products, and so on and so forth. So this was a very particular example, but it was a nice case study. And we also found that if you remember those high-throughput assays that I was discussing, where you can see if a chemical is interfering with a biological pathway. Now again, this doesn't necessarily mean that it's toxic, but it means that it can interfere with a pathway. We found that using the product intake fraction in some cases, and also using normal cosmetic use scenarios, we found in some cases people's use of cosmetics can lead to exposure that's higher than what could elicit a bioactive response. One of the examples of chemicals in this specific study was caffeine, for example. So we know by the amounts of caffeine that most of us are consuming every day that it's not a particularly toxic chemical, but it does interfere with the biological pathway. So it's important to note with these screening assays, it doesn't necessarily mean that there's a toxicity endpoint, but in general, the assays are designed to try to elucidate these potential toxicity endpoints. Now I'm going to switch gears, take a sip of water from this lovely glass water bottle, and switch gears to talk about food packaging. This food packaging work led to two different studies. One was a policy analysis, and one is a modeling paper. And I really like the food packaging work because food packaging right now, I don't know if any of you are familiar with the New Plastics Economy report, but it's really a hot topic right now in terms of circular economy and recycling and resource use, and also moving towards a bio-based economy. So if we want to make bio-based plastics and all of these different things. And then there's also been some recent studies, for example, detecting higher amounts of fluorotelomeres, which are a pretty controversial and potentially very toxic chemical in bio-based plastics, because my hypothesis for why this is because and this is based on a conversation with a polymer engineer, is that the recipes for making bioplastics are quite new. And so when they make these bioplastics, they don't necessarily have the same properties as the polymer-based plastics, and then they need to add different chemicals, additives, to try to kind of compensate and create a product that has the properties that's desired by the consumer, or by the user of the purchaser, basically, of the plastic. So with bioplastics, this is really interesting. With circular economy, it's interesting because if you mix all sorts of different plastic recycling streams, then you can imagine, and then make a food package, you can imagine that there's chemicals ending up in that food package that weren't necessarily regulated or supposed to be there. So just a very interesting case study where environmental sustainability kind of intersects with chemical exposure. And there's no methods currently to try to quantitatively compare this. So this was really my goal, was to set out on this mission to try to develop methods so we can quantitatively compare exposure to chemicals in food packaging, and also along with all of these other environmental impacts that are discussed with food packaging systems. To go over some of the modeling work, again, we're aiming to calculate the product intake fraction, or the PIF. Excuse me, and this is, in this case, is a function of the fraction of chemical that's transferred from the package into the food and the fraction of the food that is consumed. So this is FC, fraction of consumed, that is not wasted, and FT, the fraction of the initial mass that has migrated from the package into the food. This is on the y-axis of this figure, and this is just a generic figure. There's no numbers here, as you can see. And kind of the height of this figure, or this curve, is determined by the equilibrium of the chemical with the food, which is a function of the food, the chemical, and potentially plastic properties, although there's not enough data to know if that's true. And here, for example, I just show the water and the oil, because if you ever remember those experiments, maybe from, I don't know, when you were 10 years old, and you put the water and the oil in a jar and you shake it up and you realize that they're separating. This is because of the physical chemical properties. And the same is true with chemicals and how we estimate these chemical transfers, that a chemical could prefer to be in oil or prefer to be in water. So this will determine the height of this equilibrium. The rate of the rate until getting to equilibrium, so kind of the what this time scale will be on the x-axis, is a function of the rate at which the chemical is transferring into the food. And this is not necessarily dependent on whether it's oil or water, but it's more dependent on the temperature of the scenario. So if you microwave it, and it's a very high temperature scenario, or if it's a flash pasteurization that's quite hot, then the chemical will move faster from the packaging into the food. Also, if the chemical has a low molecular weight, if it's quite small, remember that means it also can move faster. So this is just the generic model. And towards the end of the thesis, I was having a lot of fun with making heat maps. And as also Peter said in his presentation yesterday, that no presentation should go without a heat map. So here's my presentation's heat map. And I don't want anyone to get bogged down with what's going on here, but red is high and blue is low. And red means that there's a high transfer from the package into the food, and blue means that there's a low transfer from the package to the food. The different, columns here are different times. So 60 seconds of contact between the food and the package, 2 weeks of contact, 2 years of contact. And then on the y-axis is the diffusion coefficient, the logarithm of the diffusion coefficient for 2 different packaging thicknesses. And on the x-axis is really that equilibrium value, the logarithm of the equilibrium value. And we can imagine scenarios where we can use these types of heat maps to quickly identify if a chemical will transfer into food at a high rate. So if you plot a chemical on this heat map and it plots in the red zone, then you know that your transfer is high. And if it plots in this blue zone, you know your transfer is low. So we're investigating right now ways to use these heat maps to try to quickly deliver information to people who don't necessarily want to take time to run a model, for example. Just to go over what this model is a function of. Very similarly to the cosmetic model, it's a function of properties, so physical chemical properties of the chemical, properties of the food, properties of the package, the temperature, as I mentioned, and also the time. Okay, and now this is the fun part, is when we start using this concept in an actual life cycle assessment. So again, if we think back to that one of the first slides, where in life cycle assessment, if you are doing a life cycle assessment of a plastic or a glass water bottle, typically methods now only include exposure to chemicals that are being emitted to the environment. They're not including exposure to chemicals that are transferring from the package into food. So what I wanted to do was play on this idea of, well, what if we include this in LCA to make LCA more comprehensive? What could this show us? Is it important, and can it show us anything? On the x-axis are different percentages of the legal limits allowed in food. So this is 100% of the legal limit. So all of these scenarios are safe in terms of regulation, regulatory thresholds. The 100% on the left, and then all the way down to 0.001% of the legal limit. And what I want to draw your attention to here is the ratio of or basically the relative contribution of these different bars. So the bar colors correspond to the legend on the right-hand side, where white is production. Transport is actually not shown because in this case it was negligible. The use due to refrigeration of of your product, which, sorry to mention, is to provide, the functional unit of this study was to provide 1 kilogram of yogurt, but it could really be any food item because we're looking at legal limits. So it could be water or anything like this. But to go back, sorry, the refrigeration during use, the consumption during use, which is my novel contribution to this work, and then the disposal, which can be recycling, or in this case, it's incineration. And you can see that when we're at 100% of the legal limit, the black bar is dominating all of the contribution to the total human toxicity impact score. This means that if we're at the legal limit for this, in this case, it was 20 chemicals in high-impact polystyrene, then we're dominating the human toxicity impact score of the life cycle. However, if we go down to 0.001% of the legal limit, then it's quite clear that actually production is dominating the potential human toxicity in the life cycle. Now if we look at this where we compare two different, so that was comparing the different stages of one product. Now if we look at this and compare two different products, so a glass container, for example, versus a polymer, this HIPS container. The gray shaded out region here, it will come in the next slide. Right now I want you to focus on the two bars that are quite visible. The first one is the HIPS without consumer exposure, so this would be what is measured currently in life cycle assessment, and then glass without consumer exposure, also as currently would be estimated. And again, the ratio or the contribution of these different bars is on the the left-hand side of the y-axis. And in this case, the symbols, the total human toxicity impact score measured in disease cases per functional unit, is on the right-hand side x-axis, just to show you some magnitude here. Now, if we actually go the next step and include consumer exposure, we see that, again, this use stage is due to consumption is dominating. But if we look at the red circle, we also see that in this prior case, glass was actually worse than plastic in terms of human toxicity impact score. And this is not just a finding that I found in this study, but also has been repeated many times by many different studies looking at glass and plastic. And if we include the exposure to chemicals in HIPS, the ones with data available, again, that was only 20, and there could be potentially hundreds, then the human toxicity impact score pops up of several orders of magnitude higher, and it also changes the ranking between the two products. So now we have a scenario where the polymer is performing worse than the glass with respect to this impact category. Alexi, how am I doing for time? Do you know? 10 minutes. Okay, I guess I've gone a bit fast. But in any case, one of the messages here is that this is looking at different percentages of the legal limits. So here is 100% of the legal limit. We know that this is not a realistic scenario. So in some cases, for example, a chemical can actually smell or taste bad if it's transferring into food. And this means that the producers of these products know that they can't hit the legal limit, because if they hit the legal limit, no one will want their product because it tastes bad or it smells bad. So we don't actually know what a lot of these realistic scenarios is and are. And this was one of the reasons why I was trying to play around with this policy standpoint. If we look at safe products, what could it mean in life cycle assessment? But it also really shows us that there's a huge need for future work. And this is really where the United States EPA steps in with some of the best work that we've seen internationally in terms of consolidating databases on chemicals and consumer products in general. And food packaging will be part of this. However, I don't believe it is discretized for different types of food packaging to be seen. But there are databases that should become available shortly that have more information on chemical concentrations in products. And then if we use that model that I presented in a few slides back, then perhaps we can take these chemical concentrations in the food packaging, we can apply the model, and we can try to predict how much the chemical has transferred into food, apply some kind of waste factor, and then understand the product intake fraction and the magnitude of mass taken in by consumers. So this is a pretty exciting area of future work. Also, as I mentioned earlier with this Pandora's box analogy, once you start speaking about exposure to chemicals in consumer products, you open up the floor to all sorts of questions from regulators, from risk managers who say, well, in these cases, the toxicity is below the safety threshold, so really this should be minimal. But if we look at this in the life cycle assessment standpoint, that's not how we consider toxicity, because actually the exposure to chemicals in consumer products can be much, much higher than exposure to chemicals distributed in the environment. And in life cycle assessment, we still consider this as an impact category. There's also discussions, people bring up, well, life cycle assessment considers these average scenarios, which means they consider an average adult. What about subpopulations, such as children? What about pregnant women? For instance. And these are things that are in the exposure science community are really key topics because chemicals can interfere with different developmental pathways. And if you've ever heard the word endocrine disruptor, for instance, this is a really hot topic and not at all considered in the well, I shouldn't say not at all, but not yet well considered in the toxicity methods we're using in life cycle assessment. So it really opens the box to all these different things. Also, the emphasis on my work on consumer exposure, I really don't want to draw away from the importance of occupational exposure. And this is because, for example, if we consider cosmetics, there's very few documented cases where cosmetics are causing disease. There are lots of lawsuits going on right now with the potential for talc powder causing ovarian cancer, but there aren't large-scale studies necessarily showing conclusively that chemicals in cosmetics are causing disease. However, there are many examples where chemicals in cosmetics used in hair care salons, for example, do cause disease. They cause formaldehyde releasers can cause, and high concentrations of formaldehyde in the indoor environment can cause severe lung damage, migraines, all sorts of occupational health problems. So this is an area that I would like to be a focus of future work. Also, there's two exciting potential areas where these models could be perhaps applied, and one is the PEF, which is the Product Environment Footprint Project in the EU, which is a pilot phase project going on right now that's looking at ways to standardize life cycle assessment methodology for the European Union. And there is some discussion of including chemicals in consumer products, but actually it is mostly due to occupational exposure from what I've heard. And also, there's been some discussions of potentially presenting or proposing a Use Tox 3.0 or a new version of Use Tox, which includes consumer exposure. This would need a consensus in order to actually happen, but it is a discussion that's happening right now, and I see some smiles in the room. But yeah, so this could be a very nice application of the models that I developed, and this would probably, I would guess, happen in the next 2 years or 2 to 5 years for really being able to apply many different types of consumer exposure models in a Use Tox platform that can be used by LCA practitioners. So I'm going to take that and start with some final conclusions here. So with that, Pandora's box is closed and hope is left inside, I hope. And in conclusion, the takeaway messages are that this thesis, first of all, raised awareness about missing exposure pathways in LCA. I was lucky to be able to present at many conferences internationally in this type of thing and get this message out as much as I could. I also was able to develop, to help contribute to the development of a metric and a framework for setting the stage for consumer product exposure in LCA in general. And very specifically, my work developed models for cosmetics and food packaging. And in general, I hope that this work can be the first stepping stones for ensuring that chemicals in consumer products are at least considered when we are talking about sustainability of consumer products in general. So thank you very much. I would like to thank DTU for funding this project and also sources of external funding for traveling. I would like to thank Qantas for being so hospitable and having me there for 6 months during an external stay. I would like to thank all of the co-authors of the papers that were produced by the thesis work. And last but not least, I would like to thank my QSA colleagues, my friends, and my family. And a special shout out to my friends here who have, and my boyfriend also, who has dealt with me during these these really special times. And these are just some of the flags that represent some of, well, the co-authors on these papers. And for me as an American, this has been a really fun and exciting time to be able to interact with so many different nationalities and people with different backgrounds. And last but not least, I would like to thank you all for your attention. Thank you.